and welcome to the MAPS Canada webinar series. My name is Shannon Smadilla, and I'm your host for the series, as well as a volunteer with MAPS Canada. We are extremely excited to have you join us for our 4th episode of the webinar series. Throughout the next few months, we have an amazing selection of speakers, luminaries, scientists, doctors, and visionaries lined up just for you. We've gathered some of the best in the field, 10 additional speakers to be exact, to share important information and their take on the psychedelic renaissance. For those of you joining us who are not familiar with MAPS, I'm happy to share with you a brief introduction. MAPS Canada, Multidisciplinary Association for Psychedelic Studies, is a registered, charitable, nonprofit Canadian organization and is an affiliate organization of the original MAPS based out of Santa Cruz, California, which was founded in 1986. MAPS Canada furthers its mission by providing evidence-based, honest public education about the risks and benefits of psychedelics, supporting scientific research into many aspects of psychedelics, including treatments for medical conditions, neuroscience, creativity, and spirituality. Planning, conducting, and publishing scientific research of psychedelic medicines, developing psychedelics into prescription medicines, training psychedelic psychotherapists, and working to establish a network of treatment centers that can provide this service in the context of professional best practices. For more information on MAPS, you can visit the website at mapscanada.org. The funds raised from this webinar series will go to fund research associated with various studies that are on the agenda with MAPS Canada. So some housekeeping items for our webinar today. The webinar will go up to about an hour and a half. We will start with a talk from our guest speaker today, Dr. Dennis McKenna. And after that, we'll have a question and answer section. So please, if you refer to the bottom of the screen, you can see the Q&A, ask a question. Feel free to put your questions in there. And you will also be able to vote up your questions. So feel free to vote on what questions you would like Dennis to answer. So just to take a look at who's joining us today, we have people from all over the world. Wow, it's quite a list. Shelt, BC, Utah, Santa Cruz, Prince Edward Island, Vancouver, Montreal, Calgary, Brazil, Toronto, Nova Scotia, San Bruno. We have Southwestern Ontario, Montreal. We have Peru. We also have Kitchener, Ottawa, downtown Eastside, Vancouver, New Brunswick, Guatemala, and let's see who else here. More Toronto, Chilliwack, West Hollywood, West Vancouver. So welcome to everyone who's joining us. It's great to see a couple of our speakers on here as well. We have Shuni from Guatemala and Chris Dyer from Peru. And of course, we're going to be having Dennis here in just a bit. So an introduction on Dennis. Dennis McKenna's research has focused on the interdisciplinary study of Amazonian ethnopharmacology. He has conducted extensive ethnobotanical fieldwork in the Peruvian Amazon. His doctoral research, which is at the University of British Columbia in 1984, focused on the ethnopharmacology of ayahuasca and ukuhe, 2 tryptamine-based hallucinogens used by indigenous peoples in the Northwest Amazon. He is the founding board member of the Hefner Institute and was a key organizer and participant at the Ayahuasca project, the first biomedical investigation of ayahuasca. He is the younger brother of Terrence McKenna. From 2000 to 2017, he taught courses on ethnopharmacology and plants in human affairs in the Center for Spirituality and Healing at the University of Minnesota. In 2019, in collaboration with colleagues, he incorporated a new nonprofit, the McKenna Academy of Natural Philosophy. He immigrated to Canada in the spring of 2019, together with his wife, Sheila, and now resides in Abbotsford, BC. Today, here's what we will be discussing. Most people are familiar with the major natural psychedelics, such as psilocybin mushrooms, ayahuasca, San Pedro cactus, peyote, etc. All of these have a rich cultural and historical background and the pharmacology and chemistry of the active constituents is well understood. Yet, hidden within the vast biodiversity of the planet, undiscovered psychedelics remain poorly investigated, if they are known to science at all. This talk will delve into the obscure byways of ethnopsychopharmacognosy to discuss a few more of the interesting examples that beckon to a new generation of ethnopharmacologists. It will also discuss the rationale for pursuing ongoing investigations into novel natural psychedelics. So without any further ado, please help me welcome Dennis McKenna.
Well, thank you, Shannon. Thank you, everybody, for showing up. Can everyone hear and see me? Well, until I get a message otherwise, I will assume so. So I'm very happy to be invited to present in this webinar series, and very much want to support MAPS Canada and MAPS USA in their quest. As many of you know, they're leading the charge. They're leading the efforts to bring psychedelics such as MDMA and psilocybin into the realm of biomedicine so that they can be used therapeutically. And it's a long, difficult path to take a substance which formerly was prohibited in the highest, most restrictive categories, and turn that and overcome those obstacles and turn that into a therapeutic medicine that people can use. So I have to add kudos to that and thank MAPS for doing what they're doing. They are really transforming medicine, mental health care, psychiatry, and ultimately society. I think the potential for psychedelics to have a great impact on some of our most challenging problems that we're facing on the social, medical, and environmental level. I think psychedelics are keys to finding some of the solutions. So I have a presentation to give here, and we will hope that the software works. It's not too long, but I'm going to start that now, and we can at last start this presentation. So the name obviously is Esoteric Psychopharmacognosy, Prospects for Discovery of Natural Psychedelics, Novel Natural Psychedelics. So anybody that knows my lectures, you know I like to throw around big words like psychopharmacology, pharmacology, or psychopharmacognosy, or psychoethnopharmacology. So the psycho part of this is clear. Maybe I'm the psycho, I don't know. After that episode, I feel a little psycho. But pharmacognosy, I don't know if people know what that means. So I'll tell you what it means. It is kind of an older word, not used too much anymore, but pharmacognosy is the scientific investigation of natural products, basically, not just psychoactive natural products, but any natural products. And it used to be a course that was taught in pharmacy schools everywhere. And then at a certain point, people training to be pharmacists or pharmacologists, there was a perception that natural products were not important. And so a lot of the pharmacy schools stopped teaching them. And that's unfortunate because that happened about the same time that the general public became quite interested in herbal medicines around the end of the 80s, and there was a whole regulatory change. But anyway, that's what we're going to talk about. And I'd like to say a little bit about, with this slide, a bit about the Hefner Institute. I'm a founding board member of Hefner Research Institute. And our goals are similar to MAPS. They're farther into MDMA. They've kind of focused on MDMA to bring forward into the clinic. And Hefner's concentration has been on psilocybin primarily. And we have worked with many of the primary researchers in the field of psilocybin and have sponsored clinical trials and are basically working very closely, for example, with Johns Hopkins, the psilocybin research program at Johns Hopkins University. Roland Griffith has been on our board. He's not anymore. He had to withdraw. He didn't want to create perception of a conflict of interest, but we still work with him very closely. And then the other one I'd like to speak of or just mention is the McKenna Academy of Natural Philosophy. You can go to the website there and learn what that is about. It's a nonprofit that I founded about a year ago, and we have a broad agenda, but the focus is mainly education in psychedelics. So I want to look back to the past a little bit to some events that are relevant to what we're talking about. So in 2017, I helped to organize a symposium in the UK, which was called ESPD 50. Oops, sorry. The Ethnopharmacologic Search for Psychoactive Drugs. There's that big word again, ethnopharmacology. 50 years of research. And what this symposium was it was to commemorate the first ESPD conference, which was held in San Francisco in 1967. And that was sponsored by the National Institute of Mental Health and other US government agencies. And the first conference, the 67 conference, was closed to the public. It was restricted, access was restricted. The only thing that the taxpayers ever got out of this was a symposium volume by that name, the Ethnopharmacologic Search for Psychoactive Drugs. And that was published by the US government printing office for some years. It's out of print now. But when it came into my radar at the age of 17, it was a very influential book to me because it made it clear that ethnopharmacology was a real discipline, and particularly when it came to the discovery of novel psychotropic, psychoactive plants and fungi. So in terms of my own personal history, it seemed that it was a potential career path, and I decided to follow it and eventually made it. Well, I guess I'm still on the path. But the original idea was that there was going to be follow-up conferences about every 10 years. And it never happened because the war on drugs came along, and the government became sort of embarrassed that they'd ever presented or sponsored something like this. So those follow-up conferences never happened. And I always felt it was important to have a follow-up conference, and I tried a couple of times. Finally, in 2017, everything fell together. So we were able to present a 50th anniversary symposium of the original 1967 conference. And we did that at Tyringham Hall in Buckinghamshire, UK, in June 2017. And the beautiful English country house in the Buckinghamshire area. And thanks to generous donations from many people and support, we were able to bring a stellar faculty there, and we spent 4 days presenting results. And it was quite a landmark conference. A key difference between ours and the original 67 conference is, although the number of people in the physical space was fairly small, we only had about 60 people attending, but we live streamed it on Facebook. And using that technology, some of the presentations were viewed at sometimes more than 75,000 people at a time were watching these presentations. So a couple of things. Many more people in the world now know what ethnopharmacology means, which is a good thing. And I'll tell you what it means. This is my favorite definition. Ethnopharmacology is the interdisciplinary scientific investigation of biologically active substances used or observed by humans. And it's a kind of tortured definition, but every part of it means something in the sense that it's devoted to biologically active substances, not necessarily psychoactive, not necessarily medicines, not necessarily consumed. For example, something like arrow poisons are well within the scope of ethnopharmacology. So, and it's interdisciplinary, and it's also focuses on traditional societies. In other words, the pharmacology, what you might call folk pharmacology. And so that's the definition. And then we carried out this symposium. And by the way, I'd like to mention that all of the videos that were live streamed are actually on the McKenna Academy website. If you go to the resources page, you can look at all those videos. They're still up there. Some very interesting presentations. And one of the goals that we had for the symposium was to put out a symposium volume, as the first one did. We were trying to do this in the spirit of the original. And we decided, well, as long as we were doing it, let's reprint the 1967 symposium as well as the 2017 symposium, because by that time it was well out of date, or well out of print. The U.S. government printing office it was no longer available. So we brought a collector's edition double volume set out, and then that is still available. It was printed by Synergetic Press, and you can order it from them. So if you want to read it, you can. If you want to look at the presentations on video, you can find them on the resources page of the McKenna Academy webpage. Why is this important? Who cares if we find new psychoactive drugs by following up ethnopharmacologic leads? Well, I imagine everyone listening to this has several ideas why this is a good idea, and I'll be happy to share some of my ideas. These things are important. It's important to do this work, to find new psychoactive substances, whether they're natural or synthetic, for several reasons. For one thing, structurally novel compounds can be thought of as probes for studying novel pharmacological mechanisms and novel cellular and molecular targets. Because psychotropic, psychoactive substances newly discovered, especially if it's not one of the known classes, often has novel mechanisms, and novel mechanisms suggest novel targets, potentially therapeutic targets. So that's one of the reasons. Looking for these novel psychedelics enhances our understanding of the neurochemical foundations of consciousness and potentially the neuropathology of mental illness, and can lead to new therapeutic applications, and in a broad sense can lead to a better understanding of the nature of the mind and consciousness. So there are all sorts of reasons to pursue this ethnopharmacologic search for psychoactive drugs. And today I'm going to talk about a small subset of what is out there. There are still major discoveries to be made, and I'm going to talk about just the highlights, but there are many more. And of course, there are many more that are unknown beyond that. I mean, there's probably 50 or 60 potentially psychedelic plants or fungi that are not investigated, but there may be hundreds, and it's hard to say, but they're definitely undiscovered, potentially psychedelic plants and fungi that are worthy of investigation for the reasons that I'm showing on this slide here. In addition to which, just scientific curiosity. It's nice to know what's out there. Here's an example of how ethnopharmacological investigations can pay off. Many of you are familiar with salvia divinorum, the hallucinogenic or psychoactive. I wouldn't call it psychedelic, but it's a psychoactive Mexican mint. And it's not a true psychedelic. It's a profoundly psychoactive, and it's used in shamanic ceremonies by the Mazatec and other groups in Guatemala. And the active ingredient, the major active ingredient, is a diterpene called salvinorin A. And it's unusual for a number of reasons. For one reason, it's not an alkaloid. Most CNS active natural compounds are alkaloids. That is, they contain nitrogen. It's rare that to find a psychotropic plant, a psychoactive plant, where the active ingredients are not alkaloids. Cannabis is one example. Cannabis, and an alkaloid, by definition, is a natural product that contains nitrogen. And this contains no nitrogen, as you can see, although it has a very complex structure. So it turns out that salvinorin A is an extremely interesting compound. It turns out that it is the most active, most selective, and potent kappa opiate agonist so far discovered. Most of you know that most what we call classic psychedelics interact with serotonin receptors, specifically and usually the serotonin 2A receptor, the 5-HT2A receptor. Salvinorin A is not a psychedelic because it has nothing to do with serotonin. It works on one of the three subtypes of the opiate receptors. There are three types known: the mu, the kappa, and the delta opiate receptors. The mu receptors are the opiate receptors that narcotics like morphine and heroin and these types of compounds interact with primarily. So they are responsible for analgesia, euphoria, ultimately addiction. The functions of the kappa opiate receptor are much less well understood. However, it's interesting in this diagram here, it turns out not only is salvinorin A selective for kappa opiate receptors, that's the only one it hits. The green sequence in the back, these are receptors. I don't know if you can see my cursor here, but these are receptor types on the lower axis. And you can see that salvinorin A only hits one receptor and nothing else. That's almost unheard of for any drug, whether it's synthetic or not. In the front there, you have the receptor profile for LSD. And as you can see, it's what pharmacologists sometimes call a dirty drug. Dirty in the sense that it's not particularly selective. It works primarily with serotonin receptors, but it interacts with many subtypes. And then it interacts with a number of different types of receptors. That's the more typical situation. The situation with salvinorin A, where it's highly selective, is normally not how you find it. The scientist that worked out the pharmacology of all this, Dr. Brian Roth, who is the director of the NIMH psychedelic drug screening program, worked out the pharmacology for salvinorin A. And he told me once, he said, if I had set out to design a kappa selective opiate agonist, it wouldn't look anything like salvinorin A. It just doesn't, there's nothing about the structure that would lead you to believe that it has that property. But in fact, it does. Not only is it selective, it's also very potent. Salvinorin A is actually the most potent psychoactive natural product so far discovered. It's active at less than 50 micrograms orally. So it's in the same realm of potency as LSD. But of course, LSD itself is not actually a natural product. It's a derivative of ergot alkaloids, but LSD itself is not a natural product. So the salvinorin A story is just an interesting sort of validation of my premise here, which is that if you look into the chemistry and pharmacology of these more obscure psychotropic, psychoactive plants and fungi, you're likely to make some discoveries that will go somewhere. And in fact, there is a lot of research now looking at the structure activity properties of salvinorin A and intimations that it may be good for things like depression, possibly PTSD, the usual spectrum of therapeutic effects that we normally think about when we think about psychedelics. But salvinorin A is not a psychedelic, and the state of mind that it produces is often dysphoric. It's not particularly pleasant for many people. It's challenging, but some people do like it. I guess there's no accounting for taste. But here, this is one example. Another one, which is well known, of course, to all of you, to everyone listening probably, is Taran Boga. And Taran Boga has an important role in the Bwiti cults in Gabon and Cameroon and that part of Western Africa. It's ancestral medicine. It's used in initiation rites. Young men and women of the tribe who are undergoing an initiation into adulthood chew very, very large amounts of the roots, almost lethal doses of the roots, and they undergo a 24 to 36 hour trance-like state. And in the trance, the ancestors appear to the initiate and impart the tribal wisdom and the ethics of the tribe. So it's very important in traditional shamanic and ceremonial life of the Bwiti. However, it's also an endangered species. So Ibogaine, the major active alkaloid of Iboga, which includes a bunch of other alkaloids in the same family, same structural family, ibogamine, cornaridine, tarananthine, and so on, has turned out to have various interesting applications in the treatment of addictions, particularly opiate addiction. It doesn't work only on serotonin, it works on a variety of neurotransmitter binding sites. So it's not a true psychedelic, but it certainly has been shown to have a therapeutic property in treating addictions, and especially opiate addictions. So as a result of that, there's great interest in Iboga, and Ibogaine itself, and efforts are being undertaken to get, there have been clinical trials of Ibogaine, and there needs to be more. But as a result of the interest, there's a lot of pressure on Iboga, and Iboga is not an abundant plant. It's being over-harvested. Effectively, it's now an endangered species. Turns out there are only 2 recognized species of Tabernantha. Tabernantha iboga Tabernantha is the genus. However, there is a closely related genus in the same family, Tabernae montana, and it turns out that it also is rich in this family of very complex indole alkaloids, which have come to be sort of described under the rubric of Iboga alkaloids. And there are many structural variations on Ibogaine in both Tabernantha and also in Tabernae montana, and many other species of Tabernae montana. So in Peru alone, there are 18 recognized species. These contain a great diversity of Iboga-type alkaloids. Some species of Tabernae montana actually do contain Ibogaine, but not in great quantities, but it is there. These other derivatives, these other Iboga alkaloids may have an action similar to Ibogaine, maybe even superior to Ibogaine. Ibogaine has some issues with cardiac toxicity, and some of these other alkaloids may be just as effective and not have this property. So here's a little I wouldn't call it a small project, but maybe a focused project, would be to gather together in some place as many species of Tabernae montana as one could obtain, and then sort through their molecular profiles. What you might call their molecular library. You might discover some compounds that are actually preferable to Ibogaine in terms of the therapeutic targets that are being discussed, or of interest, I should say. Here's one of them, Tabernae montana species. It's Sananho. Like almost all folk medicines, the Tabernae montanas have a variety of folk uses. They're used as analgesics, they're used as disinfectants, oral disinfectants, they're used for eye wounds, they're used to treat toothaches, they're sudorific, that is, they induce sleep. They have a variety of different activities that are recognized in folk medicine. Of course, folk medicine recognizes many different common names, not just this one. And Tabernae montana is often used as an admixture to ayahuasca. And it's used as a dieta plant. If you know the practices of ayahuasca, shamans in training to use ayahuasca will often diet with other plants. And this is one of the plants that they like to do that with. Sananho is the common name. So sorting through these Tabernae montana species might turn up some very interesting alkaloids. This is worth looking into. Here's another molecule that may be familiar with a lot of you. It's certainly not unknown. Harmine is an alkaloid, and I sometimes call it a hoary old alkaloid because it's old. It was discovered in the 1850s, and it was originally isolated from Paganum harmala, which is the giant Syrian rue, which is why it gained the name Harmine. Most of you know that ayahuasca is an orally active preparation made from Banisteriopsis caapi, a huge jungle liana, woody liana, and an admixture plant containing dimethyltryptamine. And in most parts of the Amazon basin, the admixture plant is a member of the coffee family, called Psychotria viridis is the scientific name. Chacruna is the common name. Dimethyltryptamine is not orally active unless you mix it with an MAO inhibitor, a monoamine oxidase inhibitor. It's broken down by the enzyme monoamine oxidase in the gut, and it never reaches the blood-brain barrier. That's why DMT, or dimethyltryptamine, has to be smoked or snuffed or injected to be active, except ayahuasca is a combination of Banisteriopsis, which contains harmine and other big carboline alkaloids, and these are all potent inhibitors of monoamine oxidase. So if you brew it up with the admixture plant, it will become orally active, radically changing the pharmacokinetics and the profile of the experience. DMT smoked or injected lasts very short time, maybe 20 minutes. It's very intense, but it's very short. When you take it in the form of ayahuasca, that pharmacokinetic profile is stretched out to 5 or 6 hours. And it's a less intense experience, but in some ways a more therapeutic experience because people can stay in that altered state for a longer time. And in the case of smoked or injected DMT, that's very difficult. It's so astonishing, and that's about all you can come back with unless you're paying very close attention. In ayahuasca, you can examine this altered landscape at a more leisurely pace and learn more from it. So harmine is the main monoamine oxidase inhibitor in Banisteriopsis. The others are harmaline. There's only a trace of that in ayahuasca, lots of it in Paganum harmala, but only a trace in ayahuasca, Banisteriopsis caapi. and then tetrahydroharmine, which is a whole other story that I'm not going to get into too much here. But it turns out, so we thought that the pharmacology of ayahuasca was pretty well understood. We thought the pharmacology of harmine was well understood until recently. But new data has come out about harmine, this 150-year-old alkaloid that we thought we knew all about. And it turns out it has some unexpected properties. For one thing, it is an inhibitor of BYRK1A, which is an important cellular kinase that regulates all sorts of CNS processes, including neurogenesis, the stimulation of nerve cell growth. And harmine will do that, at least in the Petri plate. It will stimulate nerve cells to grow. And neurogenesis is very important because it's one of the processes that helps form new connections and more elaborate connections in the neocortex in other parts of the brain. So harmine can stimulate that. And that's actually big news, only discovered a few years ago, and points the way potentially for its utility in treating, for example, dementias or cognitive deficits, as well as other things. It also interacts with different receptors, the 5-HT2A receptor, which is the target for psychedelics. It doesn't have a very high affinity for it, but it's respectable. The dissociation constant is 230 nanometers. And it also interacts with the 5-HT2C receptor at a much lower level. Also with some other receptor sites that we don't hear too much about. The I2R receptor, the I can't even pronounce them. It's the imidazoline receptor, and also with the dopamine transporter. The dopamine transporter is similar to the serotonin transporter. It inhibits the reuptake of dopamine, and it has a spectrum of activities. It's an antidepressant, an anxiolytic. It increases brain-derived neural growth factor in the hippocampus. The implication being that it enhances memory and may stimulate neurogenesis in the hippocampus. It's putatively a hallucinogenic, and it probably is, but at very high doses. It has anti-addictive properties. It ameliorates withdrawal from morphine and a morphine-dependent rat model. It has neuroprotective activities. Potentially, it could protect against brain damage due to stroke because it activates a glutamate transporter gene that is involved with neuroprotection in ischemic events. So here we have a 150-year-old alkaloid that has a lot of tricks, still a lot of properties that have not been very well looked into, and hopefully it will be. And this harmine is one of the simplest beta-carbolines, but like the tryptamines, beta-carbolines are pretty well distributed in nature. They're not as widely found in nature, but there are quite a few other plants that contain beta-carbolines besides Banisteriopsis caapi and Pagannum harmala. And in the naturally occurring beta-carbolines, you find incredible molecular diversity. And molecular diversity is a good thing because it means molecular diversity often points to pharmacological diversity. So there's a great number of beta-carbolines that need to be looked into for their potential psychedelic or psychoactive properties. Here's a few of them, and here's a few more. And as you can see, the complexity of these molecular structures is just incredible. Some of these larger ones, there's no way that organic chemistry would be able to reproduce these molecules synthetically. They have to come from nature. Who knows what their pharmacological activity is? A lot of these are antiviral, antimicrobial, and so on. They haven't really been looked at for potential central nervous system activity. And then here are yet a few more. So beta carbolines are not rare in nature. Many plants do contain them, so there's a rich area to investigate here. And they're templates potentially for drug design. Medicinal chemists are, what they do is they often start with a natural compound, and then they do theme and variation. They modify it and to improve on it. Sometimes they just modify it to make it patentable. I guess that's legitimate too. But often structural modifications of these molecules can change their properties. They can make them less toxic. They can change their pharmacokinetic profile. They can do lots of things. Medicinal chemists are very clever, and beta carbolines offer a template for what they call structure-activity relationships. In other words, how does the molecular structure, the configuration of a molecule affect its activity at whatever molecular targets you're looking at? In our area of interest, usually neurotransmitter, but it's wider than that. There's a wider scope than that. So these are interesting. Also, ayahuasca, as you know, it's mainly made from two plants, Bemstereopsis caapi and one of the DMT-containing admixture plants. But around ayahuasca, there's a large pharmacopoeia of what you might call admixture plants that may be used occasionally in conjunction with ayahuasca, and are often used in the dieta, which is a traditional practice in Amazonian shamanism. If you're a shaman in training, you want to learn how to use a variety of medicinal plants, you have to undergo a dieta, which is time spent in isolation, where you actually ingest some of these plants, often called plant teachers. And in that process, and with the help of ayahuasca, you learn the property of these plants and what they might be good for. So back when I was working on my PhD, back in the late 80s, I was interested also in the admixture plants and the sort of, ayahuasca pharmacopoeia, this galaxy of plants, which there's maybe 150 different species occasionally used. So my colleague, Luis Eduardo Luna, and I, we did just a literature review on some of these plants. We did a literature review on 52 of the genera that are purported to be used as ayahuasca admixture plants. And many of these were purported to be hallucinogens. That was the term at the time, hallucinogen, so psychedelic, I guess you could call it. But 23% of the plants we used had a reputation for being psychedelic in their own right, not only in conjunction with ayahuasca, which I think is pretty interesting. And of our sampling, 11% had unknown constituents. That's always exciting. If you're looking for new things, it's good to look at plants where the chemistry is not well characterized. 6% of them have indole alkaloids. That was also interesting because indole alkaloids containing the indole ring, are often psychedelic or psychoactive at least. 2% had other kinds of alkaloids. 4% had non-alkaloidal constituents. So this ayahuasca admixture collection is really an uninvestigated folk pharmacopoeia. And here's another summary of these. These are from that sampling. These are the genera, some of the genera that came up in a review that have the reputation of being psychedelics by themselves, sometimes used with ayahuasca, sometimes used by themselves in different contexts. And most of these, probably only the ethnobotanists in this crowd may recognize a lot of these genera. Some of you will recognize some, like Varrola. Varrola is the source of the DMT-containing snuffs that is used by different tribes in the Amazon, and so its chemistry is well understood. Occasionally it is used as an admixture plant, but the others are virtually unknown, both in terms of their chemistry and what the active constituents might be, and also what their pharmacology might be. So there's work to be done here for a couple generations of graduate students. The problem is not that people don't want to work on these, it's getting the funding to look into this. And then there is also, in some ways, it's a race against time, because the habitats are threatened, the species are threatened, and the traditional knowledge is disappearing. So this is the reflection of folk knowledge, and the folk knowledge is, it won't exist unless someone records it, unless someone writes it down. Many of these tribes, they don't do that. They don't think of that way. So this knowledge of these plants and their psychedelic properties is stored in the heads of medicine men. And it's not written down, medicine men and women, I should say, because women are also involved in ayahuasca shamanism. But Mark Plotkin famously said, when a medicine man dies, it's as though a library has burned to the ground. And that's actually true when you think about it. There is vast knowledge stored in the memories, in the experience of traditional healers. When they go, that knowledge goes, and who knows what is sacrificed. So this is another reason to preserve these plants and try to preserve the cultural integrity of the habitats of the shamanic traditions. Here's another interesting admixture plant that came out of this work. This is called Toé negro, and it's an Acanthaceae Teleostachya lanceolata. It's a very common plant around the Iquitos. You can even find it growing in the trophic islands in the Iquitos if you're crossing the street and don't get killed by the motor carros. But this is an interesting plant. It's used occasionally as an additive to ayahuasca, but only used in witchcraft. The Kokama Indians are said to use it directly as a psychedelic. They say when it's used alone, 10 leaves are gently boiled for 7 hours. The intoxication may last for 3 days, during which there is a conversation with the plant. It may cause loss of sight for up to 3 days. Who wouldn't want to take this and see what it's all about? I don't know. Not me, personally, but it's interesting because the chemistry of this is a complete mystery. There has been no investigation of what the active principles might be. Here's another interesting one from the same folk pharmacopoeia, the Coropita guianensis, often called the cannonball tree, or in the Amazon it's called Ayauma. And it has many folk uses. Its bark is used as an admixture to ayahuasca. It requires a 30-day diet. It's used to treat strong sicknesses and to fortify the body, as it says. Its spirit is said to have no head. The fruit juice is used to exorcise witchcraft. It's used for toothaches, dermatoses, dysmenorrhea, and there are unconfirmed reports that the rotting fruits are infected with highly hallucinogenic fungus. Well, I haven't bioassayed the fungus, but I have seen fungus in many rotting fruits because these things tend to drop to the ground and they split open, and they're already moldy inside before they even split open. So that fungus may be very interesting. I probably won't bioassay it personally, but then that's what you have graduate students for. Sometimes younger and healthier and maybe more reckless. I don't know. This is one worth looking into. So what else is out there? I've talked about some of the known ones. and some of the ones associated with known psychedelics. But what else is out there? Well, potentially, it's hard to say because we're talking about an unknown quantity. There may be a few dozen, there may be hundreds, and we can't really know that until it's investigated. But here's one that just surfaced in the last couple of years and was actually reported on for the first time in the ESPD 50 conference that I told you about. It was discovered by one of the presenters, Dale Millard, in an expedition to Mount Mabu in northern Mozambique in 2017. It's a pretty common looking plant. It looks like a vetch. It is a legume, and it doesn't look particularly remarkable. The plant, which I can't pronounce, Aeschynomene, I think, cristata, has many medicinal uses in Africa. And there are a number of species of this genera with pretty much a worldwide distribution, but primarily in Asia. It's used for many medicinal purposes, but this is the first time its use as a psychedelic has been reported. And Dale's informant told him that they prepare a snuff from the powdered root bark and they insufflate it to see the small people, according to an informant. That's very interesting because Varroa snuffs are used in the same way. And the chemistry of the active constituents here are unknown. If I had to guess, I would say it's probably another tryptamine, because probably DMT or 5-methoxy, as Varroa is. So maybe the compound isn't new, but there's no way to know. So it's worth looking into. It may prove to be disappointing, just another tryptamine, but you don't know what you're going to discover until you actually make an effort. Here's another vast area of molecular diversity that I'm sure will yield some new psychedelics if we're able to look into it. These are fungal endophytes. Endophytes are fungi that live inside of plants but are not pathogens. They're symbiotes. And they're very common in grasses and they're actually probably present in all plants. But in the grasses, we have a number of fungi that are very rich in ergoline type alkaloids, which are shown here. Ergolines are lysergic acid derivatives. This particular plant, Achnoptera inebrians, drunken horsegrass, you can tell from the common name that it is psychotropic, psychoactive. And the endophyte within the drunken horsegrass, Neotyphodium guacensis, I guess, produces a spectrum of ergoline alkaloids. And many other grasses and sedges have similar fungi in them. So you have, again, a rich area of chemistry, the ergolines, which we know enough about that group that it's reasonable to suppose there may be some new psychedelics in that group if effort could be made to sort through these molecules in terms of characterizing their chemistry and their pharmacology. Another novel place to look for possibly new psychoactive substances is fish. And there are a number of species of psychotropic fish that have been reported for years. And some of these, the so-called dream fish of Norfolk Island in the Pacific Ocean. National Geographic photographer tried the fish, described the dreams as pure science fiction. Sounds like that's worth looking into. Interesting things about these fish. They are sometimes consumed as food, and they're not psychedelic or psychotropic. So it seems to happen on a seasonal basis. One of the ones that's come to light recently, got attention recently, is the Salima porgy, Sternopteryx species, called the fish that makes dreams. It was used by the Romans as a recreational drug back when the Roman Empire was at its height. It's also been reported to be used in ceremonially in Polynesia. And the effects do not sound like something you might take as a recreational drug. The effects are said to be demonic, not very pleasant, and they last about 3 days. One of the informants, one of the more recent informants, reported that he went to a ceremony in which this fish was served with the intention of having a psychedelic, psychotropic experience. And sure enough, it delivered. And as he tried to drive away from the ceremony, he was being chased by 30-foot tall tarantulas, which hopefully were not real, only in his imagination. But you gotta look. Apart from why would we go looking for something like this again? The point is that whatever it is that does this, whatever the active ingredient in these fish is, it's almost certainly a new compound, given its source and given some of the reports on the activity. So again, it's worth looking into, because you don't know the value of what you discover until you actually discover it. And there may be derivatives of this that could be developed as therapeutic compounds. So this is not uncommon. There are worldwide reports about these psychedelic or psychotropic fish. Another potential source, and surprising there aren't more of them, are insects. Given that insects and plants have a very close symbiotic relationship that's all mediated through plant and insect chemistry, and insects often sequester secondary products in their larval stages to use as protectants or repellents. And so there are very close associations, and not a lot of reports of psychedelic insects or psychotropic insects. I did find one, and this is a moth, the Myelobia smirintha, in Brazil, called the bicho de taquera. I don't know what that means, but this was reported by the French explorer Saint-Hilaire, who traveled in Brazil in the early 19th century. He reported that the larva of this moth, which feeds on common bamboo, was consumed by the Malilas Indians, a tribe native to the Brazilian province of Minas Gerais. When the intestinal parts are eaten, it induces fantastic dreams lasting more than a day. Needless to say, there's been no contemporary confirmation of this report or any chemical investigation. So this is another large area where some good old pharmacognosy could yield some spectacular results. Another one that keeps coming up is Belitas. There are various Belitas species. Belitas, as you know, they're mushrooms, but instead of having gills, they have pores on the bottom. Many Belitas are quite delicious to eat, but they're also psychedelic species reported. There's an interesting cultural phenomenon in New Guinea. The Kuma people of Papua New Guinea engage in a cultural event, I guess, cultural tradition called mushroom madness. They, at a certain time of the year, they collectively eat these mushrooms, and they cause a sort of a state of madness characterized by manic behavior. And these Nanda are known, various species are implicated in this practice, especially Belitas manicus. There's no chemistry to explain this, and this may be a cultural practice, not a pharmacological practice. People have looked at the Nanda Belitas and found nothing to indicate what the active ingredients might be. So it's a report that's easy to dismiss. But then a few years ago, a well-known mycologist, David Aurora, reported on a similar phenomenon in China. He reported on the so-called Jiao Ren Ren mushrooms, the little people of Yunnan. And people consume in China, especially people consume Belitas as food, as anyone would, because they're quite delicious. But some species, when undercooked, cause colorful hallucinations of tiny people, animals, or insects. So that's why they're called the little person. And these are blue staining Belitas. And as we know, psilocybin mushrooms also stain blue, but this probably isn't psilocybin. The descriptions of the effects, at least, are unlike psilocybin. So it may be that there are novel compounds in these mushrooms that are similar to psilocybin, maybe not at all similar. I had some friends who were very excited about this, and they wanted to go to China and test this out for themselves. And as it turned out, the person that did this work speaks fluent Chinese. So they went to all the mushroom markets in this area during the mushroom season. They ate all the Belitas that they could find, and they had absolutely no effect. And the people were not very helpful. They didn't really even relate to this as a psychotropic mushroom. But hard to say. They were not successful. Yet there are other reports from Westerners that if you get it right, it seems to have something to do with not cooking them well enough. Then if you get the right species, this property shows up. So here's another area in the fungal realm, as well as the fungal endophytes. Here's a few years ago, I got a grant from the Stanley Medical Research Institute, which supports research on schizophrenia, trying to find better medicines to treat schizophrenia. And for somehow, I got a grant from this group. They said that they wanted to look into the Amazonian ethnopharmacology for potential new compounds to treat schizophrenia. And I applied for that grant and got it. What we did, we surveyed we used receptor binding, radioligand receptor binding as a screening tool. And we screened about 150 different fractions from maybe 300 different plants that for one reason or another had indications of CNS activity of some kind. And, we got some very interesting results. We did not find the novel chemistry what we were looking for, but we did find some interesting species that had not been looked at before from this respect. And we looked at, as it says, 300 species, 300 extracts from about 130 species of Amazonian medicinal plants at 31 different neurotransmitter binding sites. And as we know, the psychedelics, the true psychedelics, have the strongest activity, interact most strongly with the 5-HT2A and 1A binding sites. So this just shows out of that sample that we looked at, these are the ones that had the highest activity for the 5-HT1A and the 5-HT2A receptors. So this sampling of about 10 different genera, different species. No, one of them is Tabernae montana. That would be what I would focus on for the next phase of this work, to begin using the receptor assays to try and isolate what are those 5-HT2A active compounds and what are their pharmacological activities. So, okay, so that is my talk, and I am really sorry for the really sorry for the technical glitches, but that's the way it is.
No problem. We're just really happy to have you here, Dennis. So are we okay to go into some question and answer here? We have a few questions from our viewers.
Sure.
All right. Most popular, we'll start with the most popular here right on the Crowdcast. And the question is, what do we know of the anti-inflammatory properties of psychedelics? What are the potential new applications that could benefit from such properties?
Well, what I know of it, and what many people in this audience probably know a lot more, some of these psychedelic phenylisopropylamines, these synthetic psychedelic amphetamines, they're called amphetamines because they have that structure, but they're definitely serotonin active compounds, and many of them have strong anti-inflammatory effects at ridiculously low concentrations, far less than any detectable psychoactive or psychedelic effect. At picomolar concentrations, these things are very strong anti-inflammatories, and many of the Shogun collection of these compounds have that property. And the person that's looking into this and actually doing the work instead of just talking about it like I am, but the person that's actually doing the work is Charles Nichols at the Tulane University. Chuck Nichols is David Nichols' son, and he's also a pharmacologist and a medicinal chemist, and he's carrying the torch, and he's publishing really interesting work on anti-inflammatory effects of some of these psychedelics. So that's about all I know about
Wonderful.
this.
Charles Nichols.
Charles Nichols. Go to PubMed, and there will be a lot of publications come up. Does everybody know about PubMed?
Would you like to share?
Well, yeah. I mean, I think those of you in the biomedical world, PubMed is a major resource for anything in the life sciences. It's National Library of Medicine database on all fields of life sciences, medicine, pharmacology. There's a lot of ethnobotany in there. And if you go to the PubMed website, it's totally open, free access to anyone else in the world. In my opinion, one of the best things that the US government does is spend money on PubMed, making this information available. They do a lot of other bad things with their money, but this is actually one good thing that your taxes pay for, kind of like the ethnopharmacologic search for psychoactive drugs. So check out PubMed and do searches on topics of interest. For example, put in ayahuasca and see what comes up. You'll be amazed. Or psilocybin. There's a tremendous amount of data there, and much of it's open access. Much of it isn't. Much of it's open access. So yeah.
Wonderful, thanks. Definitely something to look into. So
Yes.
We have another question here, and it's the second most voted on Crowdcast. A mutual friend we have in the UK told me about a kind of prophetic, scary ayahuasca trip you had that seemed to point at a difficult time for humanity like we're going through now. Could you talk about that experience and your thoughts on getting through this period that we're in?
Well, yeah, not just one scary experience. I had a lot of scary experience with ayahuasca, particularly sometimes with mushrooms, but in which, the experiences are very dark and they're very apocalyptic. And, I think we're all concerned with the fate of the planet and what's happening, and they just come up in the experience. And, they're scary, but they're also necessary. I think this is part of what psychedelics are doing, particularly the plant psychedelics. They're trying to reach out to us. I say often in my talks, these are messengers from Gaia. These are plant teachers, messengers from Gaia, and they're trying to get us to wake up. What do we need to wake up to? We need to wake up to the fact that we're out of sync with nature. We're out of harmony. And so we're not valuing the nature the way we need to preserve it. And as a result, we're destroying it. And I think psychedelics send a message to many people maybe not everybody, but to many people, that this is not sustainable. This needs to change. We need to reframe our understanding, our relationship to nature, and realize that it does not exist for us to dominate or exploit or destroy. We're busy doing all of those things, and we now see the effects of that, the consequences of that. So I think these dark apocalyptic ayahuasca experiences are just another way that it is sending this message. You know, I mean, it's not pleasant, but then we're in a dire situation that, you know, we don't have a lot of time left unless we get our act together. So maybe ayahuasca is trying to, you know, deliver a stronger message, a more scary message. And then we have COVID, which comes along and in a completely different part of nature, but in some ways delivering the same message. You know, that as a species, we need to slow down, we need to pull our elbows in, you know, and that's been going on for a while, and we've seen environmental benefits as a result of that. So just because, you know, I think these experiences come up because these messengers from Gaia are trying to get us to pay attention.
Exactly. And speaking of the messages and messengers from Gaia, there's a question here specific to psilocybin mushrooms. And it asks, what is your opinion as to the nature of the consciousness of psilocybin mushrooms? And are they ultimately benevolent and here to help?
Are psilocybin mushrooms ultimately benevolent? Well, yeah, we'd like to think so. I mean, they appear to be benevolent most of the time, you know. And I'm a believer in plant intelligence. I'm a believer in fungal intelligence, but I believe in it. I think we have to realize that these things are intelligent and they are conscious, but not in the way we are. You know, I mean, they don't have complex nervous systems. That doesn't mean that they can't be conscious. You know, what we're finding as we look at levels of organization is that brains are overrated. You know, and something like mushrooms, which are complex organized networks, that may be all that's necessary to have a kind of consciousness. Whether the message is benevolent or not, I think is up to the individual experiencing them and your interpretation. You know, you may have, at times you may have mushroom experiences that are not so pleasant, just like ayahuasca experiences are not always pleasant. But I think it's a mistake to attribute benevolence or malevolence to the medicine itself. That comes from us, you know, our interpretation of what we undergo. You know, sometimes people say there's no such thing as a bad trip. And in a certain sense, I believe that, even though I don't want to have bad trips, I don't seek them out. But when they happen, you can see that there's usually a lesson there. There's usually a reason for the bad trip. So the more difficult, challenging experiences can also be the most beneficial. You know, and so I've often say that, like with ayahuasca, I think the same thing applies to mushrooms. You know, that, you know, often ayahuasca, and again, I realize this is a projection, but often it will present itself as a feminine personality, and usually an old wise woman, kind of like your grandmother. You know, a wise feminine entity, and kind of like your grandmother, it won't hesitate to hit you upside the head if it thinks it needs it. It thinks you need it. So maybe that's where the unpleasantness comes. But if it whacks you upside the head, chances are you needed that. You know, and it's done with your best interests at heart. The way your grandmother, you know, she loves you, but she'll whack you upside the head. You know, if she thinks you need a wake up call. I think sometimes the psychedelics have the potential to do that too. Sometimes we need a shock. We need a way to shock ourselves out of this default mode network that we inhabit most of the time, which can get very comfortable, and is a barrier to self-reflection and a barrier to new perspectives. And that's maybe the whole point of psychedelics in some ways. And one of the main therapeutic properties is they let you step out of this reference frame temporarily. And I think that has a lot to do with its therapeutic effects. Just being able to look at your situation as though you've stepped back from it a little bit, so you have less emotional investment in what you're going through. And that can be a valuable lesson. So, I don't judge the quality of these experiences that way. I say they're all valuable. They're all opportunities to learn. That's the thing. And there's a lot of debate these days about what to call these things. Psychedelics as a term is now back in favor. Although it's been dismissed for a long time, it's like too much association with the 60s and so on. But I like psychedelic because it's the closest to what they actually do. They manifest the mind. That's what psychedelics means, mind manifesting. So I prefer that term over, say, entheogens or hallucinogens or psychotomimetics or any of the plethora of terms that is associated with these compounds. It seems psychedelics will fit if you are careful to define that. What you mean by that is it manifests the mind. And you can't say much beyond that. It manifests the mind in many different ways, depending on those important variables, set and setting, which everybody is familiar with. So I don't need to belabor that, but that's, and sometimes I think, I've been rereading Terence's book, The Food of the Gods, and toward the latter part of it, he talks about some of the terms, and these things used to be called consciousness expanding medicines. And that's also a good term. Maybe we should start talking in those terms.
Mm-hmm.
Terminology is always a problem with these things because they can do so many things. So, unlike other psychotropic, psychoactive drugs, which you can kind of nail them down, they're stimulants or sedatives or, analgesics or whatever. They have a narrow, anything can happen on psychedelics. It all is depend on you, the set. You bring the set to the party, to the occasion, and then the setting, which needs to be carefully organized and carefully chosen to help optimize the experience.
Wow, thank you. Such important information. And I know you speak to psychedelics, meaning mind manifesting. In a lot of, I guess you could say, yogic and tantric studies, the part of healing is actually getting past the mind. What would you say in regards to psychedelics working in that regard?
Well, I don't think you really can get past the mind in a certain way, even in the yogic and tantric other meditative traditions. You can get past the ego, and psychedelics can certainly do that, and yogic practices and meditation can certainly do that. But I don't think it's really possible to get past the mind, because the mind is all there is. We are living in a hallucination that our brains construct. They construct a model of reality, not reality itself, a model of reality. And a lot of what the brain does is take information in from the outside and combine it with memories and associations and sort of internal processing and create a model of reality which is comfortable to live in. Because if we actually experience reality unfiltered, we would not be able to deal with it. A lot of what the brain does is exclude things. There's actually a term in neuroscience called neural gating. A lot of what the brain does in terms of creating this essentially the movie that you are the producer, director, and star of is they selectively admit information from the outside and combine it with internal things and create this artificial reality which you inhabit, which more or less, we hope, reflects real reality out there, but we can't even say that because we are forever in our, in the prison of this reality hallucination, which is another way, really. It's an alternative term for what's now for the default mode network. It's the same thing. I think my term is more exciting, maybe more accessible to people than default mode network. But one of the things that psychedelics reliably do, they temporarily disable these neural gating mechanisms. They demolish the default mode network on a temporary basis. And that allows for reorganization, restructuring, getting out of your box, literally getting out of your reference frame, looking at things. And the default mode network, and the neural gating mechanisms, they're resilient. They always come back up. It's literally like rebooting your computer. You reboot your computer and it comes back up after a while, as it did today. Eventually it came back, and usually it works better. It gets rid of some sludge or glud or whatever they call it, which tends to build up. We tend to build up behavioral patterns, habits, ways of thinking that are not useful. So it's like a psychic scrub in some ways that makes your motor run more smoothly.
Like a defragmenting.
Yeah, exactly that. I think the analogy is very close.
Perfect. Do you have time for a couple more questions? We have quite a few in the crowdcast here. We'll switch gears a little bit back to, let's see here. Let's go to this one. What is the most interesting novel psychedelic drug you are watching currently?
That I am watching?
Watching, working on, studying.
Well, I can't really name one. I'm kind of an old-fashioned person. I don't consider myself a psychonaut. If by psychonaut you mean someone that wants to try every possible psychedelic that they can get their hands on. That's a younger person's game, and it's fine for people that, if that appeals to people, more power to them. I tend to see my relationship with psychedelics as not putting a pin on the map for every molecule that may exist in this universe, this molecular universe, because there are hundreds of these things. And even Alex Schulten, in a lifetime of work, was not able to test them all, although he tested a great many. My relationship is I tend to form alliances with trusted plant teachers that I've used, and there's rather than variety, there's depth. There's a whole lot that they still have to teach. And my plant teachers are ayahuasca and mushrooms mostly. And I've had relationships with them for as long as I've been in this game, I guess. Well, and those are the one. I just feel like there's, I've been seriously involved with these things for almost 45 years, maybe longer, and I've just learned a tiny fraction of what there is to know. So when you say plant teachers, they are plant teachers. They have a lot to teach, and we have a lot to learn. That's one of the main messages that I get from psychedelics, is a reminder of the limitations of our own knowledge. It's very easy to get a little arrogant because we think that we know more than we know, and our science encourages this to think oh, we're scientists, scientists done marvelous things. No, we pretty much have all this figured out now. There are just a few T's to cross and I's to R. And I totally disagree with that. I think it's important to always remind ourselves how little we know compared to the vast universe of what there is to know. And I think psychedelics, that's a big lesson that they teach.
That's such great advice. So how many different psilocybin-containing mushrooms have you tried? And what would you say, which species did you find to provide the most rewarding experience?
Again, it's not really a fair question. I've only had a few different species. Psilocybe cubensis, of course, is the main one, and that's what most people's main one. I've had the chance to take a Psilocybe cyanescens, one of the Pacific Northwest species, more potent than cubensis, much stronger, but basically psilocybin, and the other species I haven't really explored, to be honest. I've heard that Psilocybe azurescens is even more potent than cyanescens, but I haven't personally experienced it. I have heard that one can cause a temporary paralysis, and some people who have taken it report that. Now, I'm not sure why it does that, and it may be kind of hard to separate an actual paralysis from, an inability to move just because you're so damn loaded, that you're not either not able to or do not desire to move. I wouldn't see that as a big red flag or safety consideration, but azurescens is, I think, one of the most potent ones so far described. Me, I only really have experience with those two species, and cyanescens much less than stropharia or Psilocybe cubensis.
All right. Well, thank you. So we're almost at the end of our time here, but we have one more question, if that's okay.
Sure.
From all of the work that you've done, I'm sure you have some interesting stories. Are there any short versions that you want to share with us today?
Well, the problem is short, short versions. This is a kind of question I should have it in advance. Well, not really. I mean, I guess I can tell you a story. It's not too short, but it's kind of interesting, and it's an example of kind of the magical world that surrounds ayahuasca, particularly in a traditional context or among people that use it, which is a few years ago, I was down in Pucallpa with my friend Eduardo Luna, and we went to an ayahuasca ceremony. We had been seeing a shaman there named Don Fidel, and one night we went with Don Fidel over to his uncle's to drink ayahuasca. And his uncle lived in a very modest, very humble adobe shack, very, very, very modest home. And we were in the room, and we were taking the ayahuasca. And it was not particularly strong, or I didn't feel like it was particularly strong. And I went outside to get some fresh air, very stuffy in that room, and it was a beautiful full moon, and it was a moonlit night. And so I went outside to have a cigarette and just take in some fresh air. And I noticed that the moon there were shadows cast against the wall of this house by the bright moon, and it appeared to be a picture of a young girl in a veil with her hands folded in an attitude of prayer. you know? And I thought, no, that's really interesting. And I actually went back in and I got Eduardo and I said, Eduardo, am I stoned or what? You need to come see this, you know? And he came out and looked at it and he remarked, he said, yeah, that's really amazing. I see what you're talking about, right? So we didn't think too much more about it. We finished the trip, we walked back home, we went to bed. The next morning, we got up and we went over to Pablo Amaringo's house. I had been there. I had just introduced Eduardo to him a few days before. We went over to Pablo Amaringo's house, and he informed us that the daughter of his cousin Francisco Montes had died the previous night. And had diarrhea due to cholera, intractable diarrhea. They had taken her to see Don Fidel, as anybody in the neighborhood would, to get a remedy for this. Well, Don Fidel was away; he was with us, and so they couldn't find him. They took her to the hospital. They were not able to do anything, and she passed on. She was about 5 years old, and he said there will be a wake. There will be a gathering at Francisco's house this evening to celebrate this little girl's life, and you have to come. And we said, of course, we'll come. So when the time comes, about 6 in the evening, we came, and there in the living room, she was all laid out in a white dress, white veil, hands folded, exactly the vision I saw about 12 hours earlier, around 3 in the morning at the end of the ayahuasca trip. And, you know, I was just blown away. You know, I was completely blown away. And you know, it appears that she died at about the time I was looking at the wall in the house of Don Jose. And so it's just one of these odd things, you know. Ayahuasca, especially in the traditional context, puts you into a universe of magic and synchronicity and these sorts of things. These sorts of things happen all the time, but that was one that really, really stuck with me, you know, and kind of gives you the perspective on what these medicines are for these people, you know, who are not white gringos from other places. That this is their culture. This is the world of magic and mystery that they inhabit all the time. So for them, you know, this was not remarkable. For me, it was really a profound experience.
Wow, thank you for sharing that. That's definitely profound, and you know, just reminds us that there's more to the world and more to life than just, you know, this material dimension that we all think we only exist in.
Yeah, well, if psychedelics don't teach you that, don't convince you that there's a lot more to be experienced than ordinary reality, then you're not paying attention. I mean, that's the whole point.
Yeah,
And
exactly.
I think there are many realities, and the thing is, they're all real in some sense. We have to think carefully about the way we bandy these words around too. It's very easy to, you know, not be conscious of the way we use words like real. Well, what is real? Inside and outside, what are those things? You know, these are all functions of this artificial reality we create for ourselves to make sense of things, but they don't really have meaning beyond that. You know, I mean, there is no inside or outside. Psychedelics teach you that. You know, and what is real? Well, I say, you know, what do you mean by real? You know, and in some sense, all we have is experience. So anything you experience is real in the sense that it's experienced. Does it correspond to something out there? Again, using this term "out there" in a careless way. Maybe it does, and maybe it doesn't. But it's experience, so to that extent, it is real.
Exactly. Well, it's been such a pleasure to have you here, and we thank you for all of your wisdom and all of your knowledge. Before we sign off here, is there anything else you'd like to say in closing?
No, I would say I would urge people to visit the McKenna Academy website. That's www.mckenna.academy. And you can look at those videos from the ethnopharmacologic search. And also, we're building the website. We're adding things into it all the time. So keep an eye on that. We'll be doing more virtual events like we did before. And that's going to be our platform, since we're not able to do retreats and physical conferences and this sort of thing. Hopefully, we'll be able to circle back on that, but probably not before the first of the year. I mean, nobody can predict what COVID's going to do, so we're just sitting tight at the moment.
Yeah. Well, thank you so much, Dennis. And again, it's a pleasure, and we're so grateful to have you here and for your support of MAPS. And we hope that we can't wait to see what you're going to do in the future.
Okay, neither can I, because I don't know totally. But thank you, Shannon, for being such a great host, and thank everybody for checking in and listening to my blather. I really appreciate it. So these are our people, right?
Yeah, absolutely. Thank you, Dennis.
Okay, thank you. Let me see.
So in closing, I would just like to take the time to thank everyone who's helped out to make this webinar possible. A huge thank you to Dennis McKenna for being here with us today. I also wanted to take time to thank our sponsors who are helping out with the webinar series. The perks page that we have for the sponsors will be coming out shortly. And thank you to the MAPS Canada webinar volunteers who have been working behind the scenes. And most importantly, thank you to all of you for being here and tuning in and supporting the series, as well as this important research. So up and coming, our next episode will be next Tuesday, June 23rd with Paul Stamets. Make sure and submit your questions through Crowdcast in advance or through the website. And our full lineup can be seen on our webpage at mapscanada.org/webinar. So we have 10 episodes remaining, and if you haven't purchased the series and would like to, you're able to up until the end of August, as all videos will be recorded. You would just log back into your Crowdcast and you can watch the videos. So thanks again for attending, and we look forward to seeing you all on the next episode. Thanks again, Dennis. Bye for now.